Welcome to this lecture on evolutionary biology. Today, we are going to explore the incredible journey from Dryopithecus to Homo sapiens. Imagine looking at the evolution of the mobile phone over the last few decades. The very first phones were massive, heavy bricks that could only make simple calls. Over time, they became smaller, sleeker, and their internal processors became incredibly powerful. The evolution of humans from early primates is remarkably similar to this technological upgrade process. Let us dive into how we became the unique, thinking beings we are today.
Now that we have set the stage, let us look at the early timeline of our ancestors. Understanding this sequence is crucial because it shows the step-by-step biological upgrades we underwent. Our story begins roughly 15 to 20 million years ago with Dryopithecus, a genus of tree-dwelling apes that serves as a common ancestor to both modern apes and humans. As environments changed, intermediate forms, like Ramapithecus, began adapting to open grasslands. A major milestone was reached with Australopithecus. They were the first hominins to definitively demonstrate obligate bipedalism, meaning they walked upright on two legs permanently. Building on that foundation of walking upright, we see the emergence of our own genus. This phase is where cognitive abilities truly began to accelerate. The Homo genus started with Homo habilis, famously known as the handyman because of their deliberate use of stone tools. This was followed by Homo erectus, who mastered fire and became the first to migrate out of Africa. Eventually, this lineage led to highly intelligent species like Homo neanderthalensis and our own species, Homo sapiens. The most defining physical changes during this time were a reduction in jaw size and a massive expansion in cranial capacity. Before we move on to the mathematics of evolution, let us understand why this history matters today. Tracing our evolutionary journey is absolutely crucial because it explains exactly why our human bodies and minds function the way they do. For instance, our transition to walking on two legs explains why so many modern humans suffer from lower back pain. Our spines were originally designed for walking on all fours. Furthermore, in the field of evolutionary medicine, knowing our ancestral lifestyle helps doctors understand modern epidemics like obesity and diabetes. These arise because our ancient bodies are mismatched with modern environments. Now that we understand the physical changes, let us look at how we measure them. While human evolution is a biological process, scientists use strict mathematical models to quantify exactly how much our ancestors changed. One of the most important measurements is how fast the brain was growing. However, we cannot simply look at brain size alone. A larger animal naturally has a larger brain, just to control its larger body mass. Instead, we must compare the actual brain mass of a fossil to the expected brain mass for a generic mammal of the exact same body size. This concept leads us directly to a vital mathematical tool. The formula you see here calculates the encephalization quotient, or EQ. Conceptually, it isolates the growth of the brain from the growth of the body. In this equation, EQ is a dimensionless number representing relative brain size. M-brain is the actual measured mass of the brain in grams. M-body is the total body mass in kilograms. The symbol C is a constant that calibrates the baseline for a normal mammal, and the exponent 0.76 accounts for the nonlinear scaling between brain and body size. If the actual brain mass increases while body mass stays the same, the EQ will increase, indicating higher cognitive capacity. Before we move on, let us see this formula in action with a concrete example. Imagine you discover an Australopithecus skeleton. You measure the actual brain mass as 450 grams and estimate the body mass at 30 kilograms. The constant C is 11.2. First, we calculate the expected brain mass by raising 30 to the power of 0.76, which is about 13.3, and multiplying by 11.2. This gives an expected brain mass of 149 grams. Finally, we divide the actual mass of 450 by the expected mass of 149. The final answer is an EQ of 3.02. Physically, this means their brain was 3 times larger than expected for their body size. So, we have mathematically proven that brain expansion occurred. This brings us to another question. When exactly did these evolutionary splits happen? While fossils tell us what our ancestors looked like, we need genetics to know exactly when the Dryopithecus lineage split into modern apes and humans. Scientists use a concept called the molecular clock. Because DNA mutations accumulate at a steady, predictable rate over time, we can compare a specific segment of DNA between modern humans and chimpanzees. By measuring the genetic differences, we can calculate backwards to find the exact time our common ancestor lived. This genetic concept gives us the molecular clock formula. Conceptually, this equation takes the total genetic difference between two species and divides it by the rate at which those differences appear. Here, the variable t represents the time in years since the two species shared a common ancestor. The variable d is the measured genetic distance, representing how much their DNA differs. The symbol lambda represents the constant annual mutation rate. We multiply the mutation rate by 2 in the denominator because both lineages are mutating simultaneously after the split. If the genetic distance d increases, it means more time has passed since the species diverged. Now that we have our formula, let us use it to date the ape-human split. Suppose geneticists find a genetic distance of 0.012 between humans and chimpanzees. The mutation rate is 1 times 10 to the power of -9 per year. We plug these values into our model. In the denominator, we multiply the mutation rate by 2, giving us 2 times 10 to the power of -9. This is the combined rate at which the lineages grow apart. Dividing 0.012 by this combined rate gives a final answer of 6 million years. This physically means our common ancestor lived 6 million years ago, perfectly matching the fossil record. Now that we have used this formula, let us understand where it comes from by deriving it. This first chunk of the proof sets up the relationship between time and genetic distance. Let t be the unknown time since the human and chimpanzee lineages split. Because mutations happen independently, the human lineage accumulates mutations equal to the rate lambda times time t. Simultaneously, the chimpanzee lineage also accumulates mutations equal to lambda times t. The total genetic distance, d, that we measure today must be the sum of the mutations from both separate branches. This intuitive setup is the foundation of our clock. Continuing from our setup, we will now solve for the final result. We established that the total genetic distance, d, equals lambda times t plus lambda times t. By combining these like terms, we get d equals 2 times lambda times t. The factor of 2 is mathematically crucial here. It accounts for the fact that both species are evolving away from each other at the same time, effectively doubling the rate of divergence. To find the time t, we simply divide both sides by 2 times lambda. This algebraic rearrangement gives us the exact molecular clock formula we used earlier, proving its validity.
So we have proven the timeline mathematically. This brings us to a visual representation of everything we have discussed. Here, you are looking at the phylogeny of human evolution. Notice the horizontal axis at the bottom, representing time in millions of years ago, from 20 down to 0. On the far left, the tree begins with Dryopithecus. As we move right, the lineage splits. The top branch leads to modern apes, while the bottom branch tracks our hominin ancestors. You can see the progression through Australopithecus, Homo habilis, and Homo erectus. Pay special attention to the skull icons. They visually reinforce the massive expansion in cranial capacity that culminated in Homo sapiens at the far right. To wrap up our lecture, let us review the key takeaways. First, the journey from Dryopithecus to Homo sapiens is defined by major biological adaptations, most notably walking upright and a massive expansion of the brain. Second, we do not just guess at these changes. Tools like the encephalization quotient allow us to mathematically prove and measure this unique cognitive growth. Finally, the molecular clock uses genetic mutation rates to accurately date when our lineage split from other apes. You have now covered the full biological and mathematical story of human origins. Excellent work today.